Dr. Haitao Guo for his presentation, an overview of HBV CCC inhibitors. Thank you very much.
Yeah, thank you, John and Roger, for inviting me to give this overview of finding cccDNA inhibitors. All we can see, we are struggling with cccDNA inhibitors. Okay, this is my disclosure. And Jiaming has given a very thorough overview of HBV life cycle. I just wanted to reiterate, the cccDNA basics. So cccDNA is the first viral product made by HBV upon infection. This is a de novo synthesis of cccDNA. And cccDNA is episomal form of HBV DNA genome. It's a mini chromosome and serves as a transcription template. cccDNA itself doesn't replicate. It's only made through conversion of the HBV genomic rcDNA, either through the de novo infection or the intracellular recycling pathway. The cccDNA exists in a low copy number. It's around from 1 to 50 copies per cell. cccDNA is thought to be stable and may survive cell division. We all know that cccDNA plays a key role in the viral persistence and the reactivation of the treatment withdrawal, also responsible for drug resistance to be carried to the progeny virus. So therefore, cccDNA is an undisputed antiviral target for achieving a cure of hepatitis infection. But unfortunately, cccDNA is not only indirectly targeted by the current antivirals. So to target cccDNA, there are three major approaches. The first one will be inhibiting cccDNA biosynthesis, maybe together with the nuke, which is a very potent replication inhibitor. So this is blocking the cccDNA formation. And for the pre-existing cccDNA mini chromosome, so either you can reduce the stability of the mini chromosome, or you can silence the cccDNA transcription, because this is the only activity of cccDNA. Considering formation of cccDNA from the rcDNA, so this is a DNA repair process. People can easily understand. The rcDNA can be considered as a damaged DNA because it has the nick gaps and the terminal, peculiarities on the terminus of the rcDNA. So those structures need to be fixed to repair this into a perfect covalently closed circular DNA. So on the bottom of this slide, this is a proposed model for cccDNA formation. It starts from the replicating rcDNA in the cytoplasm. Maybe there is a deproteination step which removes the polymerase from the rcDNA, and it prepares the rcDNA in the nucleus, in the capsid for nuclear transportation. And then in the nucleus, the rcDNA is repaired into the cccDNA by host DNA repair machinery. And in the years of studies from my lab and others' lab, so we have identified a handful of host DNA repair enzymes or factors that involved in cccDNA formation. For example, when the rcDNA get into the nucleus, there's a sensing mechanism like a UVDDB complex or the ATR-CHK1 cascade that can recognize the rcDNA in the nucleus and then recruit the host DNA repair enzymes like polymerase, exonucleases or endonucleases, to fix the terminate of the rcDNA, or maybe ligate the minor strand first to form this closed minor strand rcDNA, and then the ligase will ligate the open ends and form the cccDNA at the end. So those DNA repair factors, so a lot of them actually, we have chemical inhibitors available, and some are even used in cancer treatment. So those can be tested for inhibiting cccDNA formation. And using cccDNA formation as an antiviral target has both pros and cons. So to summarize that, so in combination with the nukes, so those cccDNA formation inhibitor, if available, they can block the de novo HBV infection, right? And also can serve as an additional layer of blocking the intracellular cccDNA recycling pathway. And cccDNA itself, if it undergoes DNA damage and can be repaired by host DNA repair machinery, so those formation inhibitor or the DNA repair inhibitors could also block the cccDNA repair. And also the inhibitors of DNA repair enzyme, and as I mentioned previously, used in cancer therapy that can be repurposed for this. But the concept for that is that there's, you're targeting cccDNA formation, there's no direct effect on the pre-existing cccDNA, right? And this most likely is host targeting, and it has uncertain long-term efficacy and toxicity issue. And also from the DNA cccDNA formation studies, we found that there's actually a redundant function among DNA repair factors. So if you block one pathway, another pathway may take over, to repair the cccDNA. And the major issue of targeting cccDNA is the longevity of cccDNA, which is the pre-existing one. They are very stable, and the previous study using the animal model or using the other hepatinovirus actually found that the cccDNA has a very long half-life in vivo. In duck and woodchuck, that could be about 2 months. And in the human, that's around like 6 months, even under nuke treatment. And a study from Maura Dandridge lab showed that a complete loss of cccDNA solely by a hepatocyte proliferation cannot be achieved in an immunodeficient mouse model without blocking the viral spread. So if you don't block the de novo infection, the cccDNA still can persist. And this study from Fabian Zulim's lab a long time ago, they measured the cccDNA half-life in the adult treated patients, and they calculate that it may take more than 10 years to completely clear the cccDNA pool in the hepatitis B patients. Maybe this is due to the incomplete inhibition of cccDNA replenishment by a less potent nuke like a adolfir. But even with the entecavir or tenofovir, this may be still an issue. That you cannot completely block the intracellular amplification of cccDNA formation. And later on, a study by scientists in Assembly Biosciences and collaborating with clinicians, they found that by monitoring the dynamic turnover of the nuke-resistant mutants on the serum HBV pgRNA, which is a marker for the cccDNA in the treatment under the treatment of HBV by nuke drugs. So the half-life of cccDNA pool in the lamivudine or this LDT treated CHP chronic hepatitis B patients actually was calculated as several months. That's shorter than than the previously thought, but still this is indicate that cccDNA half-life is long. So the cccDNA longevity is maintained by its inherent stability and also the constant replenishment from the replicating rcDNA intracellularly. So both arms should be blocked in order to decrease and ultimately eliminate the cccDNA pool, which is very challenging. And this is the example. So a study from Roche a couple of years ago, they report that they discovered a cccDNA destabilizer. It's called the CCCR08. So this compound you can see from the mini circle cccDNA or the stable cell lines all using the HBV infected primary hepatocytes. You can see the decrease of cccDNA by the treatment of CCCR08 compound. But the potency need to be further improved because you cannot completely remove the cccDNA. And also there's the mechanism of action is unclear. And also you can from this Sutton blot, you can see the compound also reduce the protein-free rcDNA and the protein-free double-strand linear DNA. So this raises a question, maybe the compound also can target the cccDNA formation or reducing those intermediates for cccDNA formation. And cccDNA, it's a mini chromosome, so it also undergoes the epigenetic regulation for the transcription regulation. So that's including, you know, the DNA methylation and the histone post-translational modifications like acetylation or methylation, all these kind of things. And in the cccDNA epigenetic regulation, the viral protein, which is the X protein, plays a key role in maintaining the epigenetic active state of cccDNA for transcription. And from this figure, you can see the X actually actively antagonists many of those restriction factors that can silence the cccDNA, including cddB1, PRMT1, and the most famous one is the SMAC5 complex. And most recently, we also identify HMGB1 as a silencer of cccDNA, but they all can be antagonized by HBX through different mechanisms. And this shows you that in the absence of X protein, the cccDNA is almost, you know, completely lost the transcription activity. And by this immunostaining, you can also see that, and also by E antigen, marrying E antigen and the pre-core mRNA in the cell. And also without the X, you can see the decrease of the activating histone marker, H3K27 acetylation, and the enrichment of the repressive marker on the cccDNA. So X protein is a very important molecule in maintaining the cccDNA epigenetic status. So when we can compare, you know, the cccDNA destabilization or silencing approach, so here are some thoughts we can discuss. So cccDNA can still be detected in a significant percentage of fully recovered acute hepatitis B patients, despite the presence of active antiviral immunity. And also in the patient, even with surface antigen loss, the cccDNA can still be detected in those patients. And the reactivation of HBV infection in these patients under some circumstance, like immunosuppressive conditions or chemotherapy, is not uncommon. So that's indicating the cccDNA is not completely removed or eliminated from those acute hepatitis B patients with spontaneous clearance. And so it might be difficult to get rid of the very last copy of cccDNA. So silencing that may be the alternative approach. And the only activity of cccDNA is the transcription, as I mentioned previously. So silence the cccDNA transcription activity may be a more realistic or practical treatment option to control hepatitis B. And considering the, you know, the importance of HBX in the cccDNA transcription, so HBX is an emerging antiviral target for silencing cccDNA. But the HBX is not an enzyme. It functions through protein-protein interactions. So to develop HBX inhibitors to silence the cccDNA. So this is one example from a Japanese group. So they develop an assay to identify compound that block the HBX and the DDB1 interaction, so that they can, you know, prevent HBX-mediated degradation of SMAC5/6 complex and then silence the cccDNA. So this is a proof of concept study. You can see they identify a compound called NTZ that can block the HBX and DDB1 interaction. And you can see the rescue of the SMAC5/6 protein in the presence of this compound under HBV infection in the primary hepatocytes. But the potency need to be improved. You can see that this is only a marginal effect on the cccDNA transcription inhibition by the NTZ compound. And recently, we collaborating with Jericho Sciences, which is a North Carolina-based company. They have a HBX binding compound called GS103. So you can also see that the compound block the X-mediated degradation of SMAC6 protein here. And also from the thermo-addition study, you can see the compound only blocks the E antigen production after the HBV entry after the entry steps. So that means it targets the post-entry step, maybe the cccDNA transcription. And this data clearly show you that, you know, this compound, without affecting the cccDNA level, it reduces the pre-core mRNA and the ratio between the pre-core mRNA and the cccDNA dose dependently. So this is give you another example. X can be targeted to silence cccDNA. And of course, people always think, you can use the gene editing approach to target cccDNA, use the TALEN, CRISPR, or this gene editing or designer nucleases to do that. So, but there's definitely a challenge people always also can think about, the delivery. Maybe it's not a bigger problem now because there's LNP, GalNAc, and exome-based delivery, and the liver actually it's probably easier for this kind of a delivery target. But the problem can those, the potency enough to track down the very last copy of cccDNA by those enzymes? And also maybe those approach can select the mutations and the quasi-species based on this sequence. Okay. And the cleavage of the integrated HBV DNA by those designer enzymes may cause the host genome instability because the sequence is the same between the integrated HBV DNA and the cccDNA. And also there's off-target effect could be considered. So, but in terms of the high throughput screening to find the cccDNA inhibitors, so this is still an underexplored area. So the main reason is that we don't have a really reliable and a good marker for monitoring the cccDNA level in the infected cells. So to overcome this, so we have been developing those stable cell line-based system using the E antigen as a marker for cccDNA. So basically, this is the inducible HBV stable cell line. So the integrated HBV DNA will support the replication on the cccDNA formation, but integrated DNA doesn't express the pre-core mRNA and the E antigen. But only when cccDNA is formed, the E antigen can be produced and serving as a marker for cccDNA. And later on, we also add a HA tag into the E antigen region, so which can increase the specificity for this detection. And this is just give you the validation of this system by the old school southern blot, northern blot, and cccDNA. And using this system, which is the first generation of this system, HepD19 cells, we have identified a compound that can inhibit cccDNA formation. Which we actually treat the compound at the same time when we induce the HBV replication and cccDNA formation. So this scored this cccDNA formation inhibitor. Presumably, this will reduce cccDNA by reducing the deep protein rcDNA intermediates. And recently, we have slightly modified the screening paradigm by using the induced cells, which already have the pre-existing cccDNA, and then we use the tetracycline 3-TC to prevent the de novo cccDNA formation, and then use the compound to treat the cells and find the compound reduce the E antigen, and then we look back for the cccDNA level and distinguish those inhibitors into the cccDNA transcription inhibitor or the cccDNA destabilizers. For a proof of concept study, so we screened the epigenetic compound library for the cccDNA transcription inhibitors, and we identify MS436. This is the BRD4 inhibitor, which inhibits the BRD4's height activity and the reader activity. And this is just validate how the compound works. It reduces the E antigen, reduces the cccDNA, and also changes the epigenetic status of the cccDNA. And this validate the target, and through the knockdown of BRD4 or use the PROTAC to degrade the BRD4, you can basically recapitulate the same effect of the MS436 compound. But the off-target effect on long-term treatment toxicity need to be further evaluated. And also, surprisingly, we found that when we inhibit the cccDNA transcription by MS436, we also see a slight reduction of the cccDNA level in the treated cells. This is also further demonstrated by a recombinant cccDNA system more clearly. So the implication that cccDNA stability may be influenced by the epigenetic alteration. So, and this is consistent with the previous study, which is the MLN compound that inhibit the the SMAC5/6 degradation, and also it reduces the cccDNA transcription, and also you can see the level of the cccDNA reduced. So maybe the cccDNA transcription and stability couple even. So most
We have
recently,
about one more
actually,
minute, Dr.
sure,
Guo.
sure. I will finish.
Please.
So we have further improved that system by replacing the HA tag by a habit tag, which is split luciferase. And using this assay, we can directly less the cell by the reagent contains a large bit and measure the habit tag, the precore protein in the cell lysid, which is more closely related to cccDNA transcription intracellularly. So and this system we are currently working together with the University of Pittsburgh Drug Discovery Institute to change this into a high throughput platform and for compound screening. So to summarize, elimination of cccDNA definitely is a holy grail for a complete cure of HBV infection. But silencing and inactivation of cccDNA is an intermediate goal and perhaps more practical to achieve a functional cure. And of course, inactivation of cccDNA may lead to this destabilization, as some of the studies suggested. A large scale of high throughput screening of cccDNA inhibitor in the cccDNA reporter system and the infection system, also in the presence of nuke or other potent replication inhibitor like the CAM compounds talked by Adam Slonick is required. So I just want to stop here and looking forward to discussion. Thank you.
Thank you.